It has 3 sodium sites inside the cell. Okay, you can see cytoplasm and ECM. That side is ECM and this below side is cytoplasm inside of the cell. So, and this straight line, this strip is bilipid layer. This is cell membrane, okay, bilipid layer, and this is one protein present in this bilipid layer. This is transmembrane protein. Here in this case, we call this as sodium potassium pump. So this protein has alpha beta subunit. Alpha protein has 3 sites, 3 sodium binding sites, where? On the inner side, and 2 potassium binding sites on the outer side. So what happens when 2 potassium molecules bind on the outside from the ECF, and 3 sodium molecules bind from the inside, to which alpha subunit of this sodium potassium ATPase pump. So what happens? The ATP gets activated, okay, and there is release of a high energy phosphate bond. Because of the provision of that energy, this alpha subunit changes the configuration. It changes the shape, the configuration. So these 3 sodium ions are moved out of the cell, 2 potassium ions are moved inside the cell. So basically both these ions move against the concentration gradient. Sodium out, potassium in. So this is the basic functioning of this sodium potassium ATPase pump. This is the type of primary active transport where the ATP is used directly. Is that clear? So other examples, as I told you, are the calcium pumps and sodium hydrogen pumps. This is the diagrammatic representation. This is sodium potassium pump, and 3 sodium and 2 potassium, once they attach, this is configurational change, the change in the configuration, and 3 sodium go out and 2 potassium ions move in, right? So what is the function of the sodium potassium pump? It is present in all eukaryotic cells, as I told you, okay, and it maintains sodium potassium concentration across the cell membrane, it maintains volume of the cell, and it causes negative electrical charge inside the cell. What does this mean? Quickly, yes. Maintains sodium potassium concentration means what happens actually when the cell is depolarized. When the cell is how is cell depolarized? Because of the entry of sodium. Sodium channels open, sodium moves in, so cell is depolarized. So this sodium which moves in has to be now thrown out of the cell. It has to be moved out of the cell. That is done by the sodium potassium pump. Okay, and it maintains volume of the cell. How does it maintain volume of the cell? Suppose this pump does not function. What will happen? Sodium will accumulate inside. Sodium will accumulate inside, so more water will go inside. Because of osmosis, this sodium which is accumulated inside, it will attract more water. So what will happen? Cell will swell and die. So this pump maintains the volume of the cell. Is that clear? And third is it causes negativity. It maintains the electronegativity, because any cell, what is the RMP, is usually negative. Say, for example, a skeletal muscle cell, minus 90 millivolts. Okay, any other nerve cell may be minus 70 millivolts. So, resting membrane potential at rest inside of the cell is negative as compared to outside. So, at rest, a cell has to be electronegative. This electronegativity this pump helps in maintaining this electronegativity. How? Once this pump operates, it throws out 3 positive ions and brings 2 inside. So every time this operates, the cell loses 1 positive ion. Is that clear? Because in exchange of 3 positive ions, only 2 are coming in. So every time this pump operates, the cell loses 1 positive charge. That helps in creating the negativity inside the cell. So this is how this pump helps in all these 3 things. 1, maintaining the sodium potassium concentration. 2, maintaining the volume. And 3, maintaining the negativity, electronegativity across the cell membrane. And this sodium potassium pump is example of primary active transport, where the energy is used directly in the form of ATP. Because the molecules are to be transported across against the concentration gradient. This is another example of calcium pump. Again, calcium is pumped against the concentration gradient. Secondary active transport, again here, but the energy is not used directly. Energy is not used directly but indirectly. Secondary active transport has two main types. What are those? Yes, anyone? Secondary active transport has two main types. Yes? Co-transport and counter-transport. What is that? Co-transport and counter-transport. So this is co-transport, also called as symport. Symport. Examples? Sodium glucose co-transport. Okay? The transport of sodium via its concentration gradient is coupled with the transport of some other substance. For example, you say in the example of sodium glucose co-transport. Sodium and glucose attached to this protein and both are transported inside the cell. So this is sodium glucose co-transport. If only sodium attaches to the protein, it will not transport. Unless both of them attach to this protein, then only this protein will transport both the molecules inside the cell. So this type of transport is called co-transport. For example is sodium glucose co-transport. Another one is counter-transport. What will happen in counter-transport? One substance will move in, one will move out. Here both move in the same direction. In counter-transport, one will move in, one will move out. Counter-transport, for example, sodium hydrogen or sodium calcium exchanger. Sodium calcium exchanger happens where? For example, when the muscular contraction is over and calcium is released back from troponin C. Once the contraction is over, the calcium is released back, and that calcium mostly is pumped back into sarcoplasmic reticulum. Some of the calcium moves out of the cell. That calcium moves out in exchange of sodium. Sodium comes in and calcium moves out. That is called sodium calcium exchanger. That's an example of counter transport or secondary active transport. So what are the examples? Sodium hydrogen, sodium calcium, and many other examples. So we have primary active transport. The classical example we discussed is sodium potassium pump. And we have secondary active transport, which is again divided into co or counter transport. Co-transport example is sodium glucose, okay, and counter transport example is sodium calcium exchange or sodium hydrogen exchange, right? Secondary active transport, energy is not utilized directly. And secondary active transport is mainly due to buildup of sodium concentration. And sodium concentration is built up because of what? Sodium potassium pump. That is active transport, where the sodium concentration is built up outside the cell, and then because of that build-up of sodium concentration, that it carries either glucose with it or calcium in exchange. The direct energy is not used in this type of transport, right? This is the example of counter transport. क्या करूं? हाँ, अच्छा ठीक, okay. So this is example of counter transport, right? Applied aspects, we have cardiac glycosides inhibits sodium potassium pump and increases cardiac contractility, right? This is the applied aspect of it. We use cardiac glycosides to increase the contractility of cardiac muscle, right? These are the other examples where you can activate sodium potassium pump or you can inhibit the sodium potassium pump. Vesicular transport we will not discuss in this session. It is usually used in the bigger molecules, and the types are endocytosis, exocytosis and transcytosis. We will discuss this in some other session. This again, it also uses energy. Okay.